Hello students, and welcome to our new Nightingale Academy. In today's session, we discuss about the molecular signaling pathways that are involved in cancer. Means during the cancer, how the pathway or how the signaling pathway will deregulate. Okay, means how this cancer affect our metabolic pathways. So we discuss this in detail. Okay, cancer develops when normal signaling pathway that regulate the cell growth, differentiation and survival and death become disturbed. Whenever the cell signaling will disturb, then it will disturb the cell cycle. Or whenever the cell cycle is disturbed, then the cell is go under the uncontrolled cell division. Now it will become a cancer. Okay. This pathway are activated by the mutation, by amplification, deletion, chromosomal abnormality and epigenetic change. These are the reasons. Now, below are the major, there are a different metabolic pathways are influenced or involved in cancer. So, we discuss these pathway one by one. First is your REST pathway. Okay. This REST pathway is normally in normal cell. It is responsible for the cell proliferation, cell differentiation, and cell survival. Okay, and this pathway is activated by the tyrosine kinase. Let's suppose this is the tyrokinase. Now your growth factor. Let's suppose this is a growth factor. This growth factor bind to this receptor. Okay. Now receptor are dimerization. And autophosphorylation. Okay, then your adapter protein will activate like GRB2 or SOS. Okay, this GRB2 or SOS protein will activate and it will activate the REST protein. And this REST protein is GTP link. Now this REST protein activate the REF protein. Now this REF protein phosphorate or active the MEF protein. Now this MEF protein activate the RIC protein. Or this RIC is enter into the nucleus and it is act as a transcription factor and it will transcribe the myo and OSH. This is the normal side. Means grabbed first, a receptor is bind. Okay. Then dimerization and autophosphorylation. Then GRB2 or SOS protein will bind. Then it will activate the REST protein. REST protein is a GTP link. Then it will activate the REF protein. Okay. It will activate the MEC protein. Then it will act the EREC protein. And this EREC is a transcription factor. It will enter into the nucleus and transcribe this myo and post. Okay, this is the normal. This is the condition in normal cell. But in cancerous cells, this REST protein is mutated. Okay. This REST protein is mutated. Whenever this REST protein is mutated, then here this KRES, HRES and NRES Now, during the cancer. During the cancer in rest, okay, whenever this rest gene is mutated by the, most of the time this rest gene is mutated by the point mutation. Okay. Due to this point mutation, this RAS protein along with GTP is, means it will arrest in this active form. Arrest in this active form. If this RAS protein is activated, then it will activate the REF protein, then MEF protein, then IC, and IC is a transcription factor. It will enter into the nucleus and it will activate the myo and force gene, and these genes are responsible for the cell proliferation and differentiation. Okay, now the cells is continuously divide, and cells undergo the uncontrolled cell division, and it will cause the cancer. Okay, here this KRAS, HRAS, and RAS. These are the different mutation form of this RAS gene. Okay, and this colon cancer or this pathway, due to the disturbance of this pathway, pancreatic cancer, colon cancer, and lung cancer, these are the very common cancer. Okay, okay, and this is the RAS pathway. Okay, means in normal condition this pathway is regulated by the help of this growth factor, but during the mutation this RAS protein is activated automatically. This is if this growth factor is present or not present. Now this RAS protein is GTP bound. In this step it will arrest, or this is the active form of this RAS protein. If this RAS protein is activated, then it will activate the REF, then MET, then IC. And this IC is a transcription factor. It will enter into the nucleus and transcribe the gene, and this gene is responsible for the cell growth and proliferation. Now this process is continuously going on. If this process is continuously going on, the cell is moved under to the uncontrolled cell division, and it will cause the cancer. A normal cancer is pancreatic cancer, colon cancer, and lung cancer. Okay. Now next pathway is your PI3K, AKT, mTOR pathway. Okay. Normally this pathway is responsible for the cell survival, metabolism, growth, or angiogenesis. Okay, normally this RTKs activate this PI3K gene, and this PI3 gene converted into the PIP2, then this PIP2 converted into the PIP3, then this PIP3 recruited the AKT. Now this AKT activate the mTOR. Now this mTOR is act as a promoting protein. It will enter into the nucleus and it will promote the cell synthesis and the protein synthesis, and this protein synthesis is very important for the growth. Okay, this is the normal condition. Okay. Okay. Now in negative regulation, P10, that is the tumor suppressor gene, it will dephosphorite the PIP3 gene. Okay, and this PIP3 is inhibit the pathway. Means this process is inhibited by this P10. What is this P10? P10 is a tumor suppressor gene. But in cancer, this is the condition of normal cell. Normal cell, RKT activate, it will activate the PI3K2, it will activate the PIP3, it will activate PIP2, then PIP3, then AKT, then motor, and it promotes the cell protein synthesis and growth. But in and this P10, this is a tumor suppressor gene that will regulate this particular path. But during the cancer, this PIP3 gene, that is the PI3K gene, is mutated. If this gene is mutated, then this pathway is continuously going on. Okay, and if this P10 is suppressed by the mutation, then again there is a no regulation, negative regulation of this pathway. Now cell is undergo the unconditional cell growth and proliferation. Okay, P10 loss means common in prostatic and endometrial cancer. Leads to the antipeptidic signal, increasing the cell survival and increasing the glucose metabolism. Okay? This is the PI3K pathway. Next pathway is your P53 signaling. This P53 signaling is known as the guidance of genome. Okay? It responds to the DNA damage condition. Okay? It induces the DNA repairing, trigger the apoptosis and causing the cell cycle arrest. Okay? In normal condition, if this gene is mutated, means 50 to 60% cancers are the uh, approximately 50 to 60% cancer are now this is the normal condition. But in cancer, 50 to 60% is cancer is due to this P53 signaling. In this signaling, this P53 is mutated. Okay, if this gene is mutated, then it will fail to DNA repairing, accumulation of mutations and resistance to the apoptosis. Okay, and if this signaling is disturbed, then it will responsible to cause a breast cancer, lung cancer, colon cancer and liver cancers. Okay, now next pathway is a Wnt and beta next is your WNT beta catenin pathway. This pathway is responsible for regulation of stem cell renewal, differentiation and tissues development. Mechanism: without this Wnt and beta carotene, degradation of APC, auxin and GSK3 beta complex. With WNT or beta catenin, it will stabilize and ensure the nucleus activation of myo and cyclin D1, and the cyclin D1 is again plays a most important role in cell cycle, in cell cycle. This is the normal condition, but in cancer condition, due to the mutation in APC and beta catenin, this pathway is always on. If without WNT, means WNT beta catenin degrade the APC, auxin and GS3K complex in normal condition. But whenever this WNT is present, then it will beta catenin is act as a stabilization. It will activate the myo and cyclin D. Okay? But in mutation, this APG gene will mutate it. If this APG gene is mutated, then this beta catenin is always bind and this pathway is completely on. Okay, and if this pathway is on, means myo and cyclin D continuously expressed, and it will cause the and whenever if this pathway is on, it's mean your cells is undergo the uncontrolled cell division. Okay, and it leads to the stem cells like behavior and controlled proliferation. Okay, next is your Notch signaling. Notch signaling is responsible for the control, differentiation, cell fate and tissue homeostasis. Okay. Mechanism means beta ligand bind to the Notch receptor. Okay, Notch notch receptor or notch intracellular domain release some enzyme. That's enzyme enter into the new some, then it will enter release some release some molecules. These transcription mechanism means first lambda or ligand bind to the notch receptor. Whenever this will bind to the notch receptor, then notch intracellular domain is released, and this domain is enter into the nucleus. Then it will activate the target gene. Okay, this is the normal condition. But in abnormal condition, this act as a oncogene or tumor suppressor gene. Okay, this oncogene is T cells, leukemia, and breast cancer. Okay, and the causes is increases proliferation and inhibit the differentiation. Okay. Now we discuss about the apoptosis pathway. Now we discuss about the apoptosis pathway. In apoptosis pathway is controlled by the BCL2 gene, or BCL2 family protein. P53 activate the pro apoptotic protein, and this protein like Bex and Bex. This Bex and Bex is responsible for the apoptosis process. But due to the mutation this Bex gene and this due to the mutation in Bex and Bex gene, this process will hindrance. If this P53 gene is inactivate, then it does not have a capability to activate this Bex and Bex gene. If this Bex and Bex gene is not activated, then BCL2 gene will activate. If this BCL2 gene is activated, it will move to the cell into the uncontrolled cell division. Okay. Same for the receptor. This is for the internship pathway. For externship pathway, this receptor ligand is bind to this receptor. The binding is not efficient. Okay. Overexpression. Whenever the overexpression of BCL2 gene anti-apoptotic cells, it will cause the leukemia and lymphoma cancer. If the loss of caspase means apoptosis and resistance. This is the apoptotic part. Now we discuss about this Hedgehog signaling pathway. Okay, this Hedgehog signaling pathway plays a most important role. It is a main signaling pathway of embryonic development, tissue patterning, stem cells maintenance, maintenance, and your stem cells maintenance and regeneration. Okay, there are three types of hedgehog. First is your sonic. This is most widely used. Next is Indian. It plays a most important role in bone growth signaling. Next is your desert. It plays a most important role in nerve development or nerve signaling. Okay. Now, in normal condition, these are the ligand. Okay. Now this ligand bind to the receptor that is known as a. Whenever this will bind to this receptor, then a protein is present, that is the SMO, that will released out. Whenever this protein is released out, a new protein or transcription factor, that is a GLI, is activated. Okay, and it will enter into the nucleus, and it is responsible for the gene expression of cyclin D, myo, and BCL2 gene. Okay, in normal cell condition, normal cycle. A protein is also present that is known as a S. A protein is also present that is known as a SUFU. This SUFU is a tumor suppressor gene. Okay. This SUFU inhibit the activity of GLI and regulate this pathway. Okay, in normal conditions, in normal cells. But in cancerous cells, if this UF is mutated, then means due to the mutation, loss of function. Loss of function means now this CUFU not inhibit this GLI and this pathways move is continuously active. If this pathway is active, then cell is go under the uncontrolled cell division. Okay. Next is this SMO is also gain in due to the mutation. This gain of function is found. If the gain of function, then this SMO is automatically released out. Whenever this SMO is released out, then GLP is activated and gene will expressed. Okay. This process will happen during the cancerous cells. Okay. And the type of cancer is your pancreatic cancers, breast cancer, colon cancer, basal cells carcinoma cancer, medulloblastoma cancer, etc. Okay. Now we discuss about the next signaling pathway that is known as a JAK-STAT. Okay. What is the next signaling pathway? That is the JAK-STAT pathway. Okay. This pathway is, what is the function of this pathway? The function of this pathway is, now we discuss about the next signaling pathway, that is the JAK-STAT pathway. Okay. This pathway is a rapid signaling cascade used by the cytokine. In the signaling pathway, we use cytokines, growth factor, hormones, etc. Means this pathway is the ligand of this pathway is cytokines, your growth factor, and hormones. Okay, and this pathway is very essential to maintain your immune system, immune response, cell proliferation, cell growth, cell survival, and cell immortality, and cell and hematopoiesis and inflammation. Okay. Okay. In the signaling pathway, there are four types of JAKs. JAK 1, 2, 3, and 4. And 2. It is a tyrosine kinase. Okay. And what is this STAT? STAT is the signal transduce and activators of transcription. Okay, that is known as a STAT. Okay, now in this pathway, first cytokinin bind to this JAK kinase. Okay, whenever this JAK kinase will active, then it will activate or this is a kinase. That's why it phosphorylates this STAT. Okay. Okay. Whenever the STAT protein is phosphorylized, then it will form the dimer. Means cytokine bind to ZEC kinase. And ZEC kinase is a kinase protein. That's why it performs the process of phosphorylation. Due to the phosphorylation, this STAT protein is phosphorylated. And whenever this protein is phosphorylated, then it will form the dimerization. And it is moved into the nucleus. Okay, then it will activate the gene expression. Gene expression like inflammation, cell growth, survival, etc. Okay. Okay. For controlling this particular pathway, a protein or tumor suppressor gene is present that is known as a SOCS, that is the suppressor of cytokinin signaling. Okay, this will bind to this JAK and inhibit the binding of cytokines. Whenever the cytokine is not bind, then this JAK protein is not phosphorylated. This STAT protein and no dimerization is no gene expression. Okay, a protein, a next molecule is present that is known as a PICE. It will inhibit the activity of this STAT. If this STAT is phosphorylated, it will inhibit the activity of STAT means no STAT, no dimerization, no gene expression. In the cancerous cells. In the cancerous cells, this SOCS is mutated and loss of function is happen. Loss of functions means now this JAK is free. Now cytokinin will bind and active this pathway. Okay, that is known as a conjugative activation. Okay, say again condition, if this JAK is mutated. If this JAK is mutated, then this SOCS is not recognized this JAK pathway. If not recognized, then not bind. These pathways moving, and this condition arises during the myeloproliferated neoplasma is mutated. Then this pathway is moving on, and cell go to the uncontrolled cell division. Okay, if this bias protein is in bias protein, that is the due to the mutation loss of function. If loss of function, then STAT protein is overexpressed. If this is overexpressed, then more the dimerization, more the gene expression, more the inflammation, more the
cell growth, mole, the survival, immortality, each and every condition is open, active, and cell go to the under cell and control cell growth and cancer. And the example is breast cancer, lung cancer, colon cancer, prostatic cancer, and leukemia cancers. Okay, this is the signaling path. This is the JAK-STAT signaling pathway in cancerous cells. In this cell, this SOCS, JAK, and ASO genes will mutated. Okay. Now, next pathway is your angiogenesis pathway. Angiogenesis pathway means formation of blood cells. Next is your angiogenesis pathway. What is the angiogenesis? Angiogenesis means formation of new blood vessels. Okay, new blood vessels from pre-existing vessels. Okay, it is tightly regulated by the normal tissues, but become deregulated and overactivate during the cancerous cells in normal condition. In normal condition, this angiogenesis process is happen in during in wound healing phase, menstruation cycle, and embryonic development. And tissue repairing. Normal this process is happen during these conditions. Okay, hypoxia bind to this HIF 1 alpha and stabilize this. Okay, then it will activate the bag expression. It will activate the bag and bag expression will start. Now we understand this mechanism. First, hypoxia, and this hypoxia activate the HIF 1 alpha and stabilize it. In normal hypoxia, this HIF 1 alpha protein is stabilized or accumulated and move into the nucleus and start the production of Vega gene during this normal condition. Whenever this expression is start, then proliferation of endothelial cells. Okay, or angiogenesis process will effect. But during the cancerous cells, okay, during this cancerous cells, this HIF 1 alpha gene is overexpressed. Whenever this overexpressed, then it is more accumulated. If more accumulated, then more move to the nucleus and more gene activation and more the blood cells is formed. Okay, this is the condition of angiogenesis during the cancerous cells. Okay, this is the another pathway. And this angiogenesis process is related to the breast cancer, colon cancer, lungs cancer, ovarian cancer, renal cancer, and melanoma cancer. Okay, this is the angiogenesis pathway. Okay. Doing the cancerous cells. Now I revise today's session. In cancerous cells, RAS pathway, whenever this RAS protein is mutated, then it will cause the cancer. Okay. Next pathway is your PI3K AKT's pathway. If this pathway is mutated, then it will cause the cancer. Okay. WNT beta carotene pathway is mutated, then it is again cause the cancer. If this Hong pathway is mutated, then it will also cause the cancer. If no signaling pathway is mutated, then it will cause the cancer. If the JAK and STAT pathway is mutated, then it will cause the cancer. P53 pathway is mutated, then it will also cause the cancer. These are the some pathway which directly affect the cancerous process or directly involved in the formation of cancer. Whenever this pathway is disturbed, then the cell is go under the uncontrolled cell division. Sometimes apoptosis process will stop. Okay, and whenever that apoptosis process is stopped, then it will cause the cancer. Okay, if the cell is go under the uncontrolled cell division, then it will again cause the cancer. Okay, and this is all about today's session. Please follow ITLS Academy on Instagram, Facebook, Twitter, YouTube, WhatsApp, LinkedIn, and also get it on Google Play Store. Thank you so much for watching my video. Please like, share, and subscribe ITLS Academy. Thank you.